In this video, we'll be talking about organic compounds, and under the organic compounds, we'll be talking about carbohydrates, lipids, proteins, and nucleotides. So before we go too far into those individual ones, let's have a talk about some real basic things. And that what we're talking about are monomers and polymers. So each type of organic compound, which could either be a carbohydrate, a lipid, a protein, or a nucleic acid, is what we consider a polymer. Now, polymers are made from individual subunits, which are the monomers. So monomers are single subunits that are used to build the polymers, like building blocks. And there's a process that's used called dehydration synthesis. And dehydration synthesis is, like the name suggests, is we're going to pull out a water molecule from there. And so a hydrogen and an OH group get pulled off. Then there's the opposite, hydrolysis. And if you actually look at the word hydrolysis, water breaking, it's a catabolic reaction that uses water to break a polymer down into small subunits. So we have this process where we're moving between monomers and they're being assembled through dehydration synthesis to being polymers, and polymers get broken down to make monomers through hydrolysis. So let's start talking about carbohydrates. So carbohydrates are made of carbon, hydrogen, oxygen, and one of their main roles is to function as basically a fuel source. Now, we can start to look at some subcategories, and we'll start with the monosaccharides, mono for one. So the monosaccharides are what all the carbohydrates are made from. So these monosaccharides have between 3 and 7 carbons, and some examples of them would be glucose, fructose, galactose, ribose, deoxyribose. They're the most common ones. And let's have a look at some of them. Now, we don't have to memorize the structure in detail. It's not a biochemistry course, but let's have a look. So you can see here we're looking at ring-like structures that are made by the carbons, the individual carbons with an oxygen right there. And let's have a look at some of them. These are called the pentose sugars. Why? Because they have 5 sides. We also have the hexose sugars, which have 6 sides. And once again, you can see that the ring is made by carbon. Now, as we look, we can see that here we have deoxyribose, and you may have heard this before because DNA is deoxyribose, and RNA is ribose. And the names give us a little clue. So deoxy tells us that something's missing. And indeed, if we actually look at the structures, we can see here, there's an individual hydrogen. Over here is an OH or a hydroxyl group. So you can see that the oxygen has disappeared, and therefore the name deoxyribose. Let's have a look at, here's glucose, of course, a very important hexose sugar. We have fructose, and these are all isomers of glucose. So we have glucose, and the isomers are fructose and galactose. Now you do notice as isomers, they all have the same number of carbons, hydrogens, and oxygens. It's just where things are located. And if you have a look at the 3 different structures and spend some time going through it, you can see that there are some differences as to where the atoms are located. Now, talking about monomers, well, if we start to attach them together, the monosaccharides, the basic sugars, we can form disaccharides. And disaccharides are formed by the union of 2 monosaccharides through the process of dehydration synthesis. So let's have a look. Let's zoom in. So here I'm going to take glucose, which is 1 monosaccharide, fructose, which is another monosaccharide, and I'm going to join them together. And by joining them together, I'm going to form a disaccharide. Now, how am I going to do it? I'm going to use the process of dehydration synthesis. So what I'm going to do is I'm going to pull off an OH group here and an H there. So only thing that's going to remain would be this O. This will be gone, and the O will remain. And let's have a look. So if I do dehydration synthesis, I pull off that water molecule. Once again, that's the OH coming off and an H. So that's two H's and an O, so it's H2O, gets pulled off. And that allows the two monomers to bond. And we can see it happening here. And that's the process of dehydration synthesis, is going from the monosaccharides to forming a disaccharide. And in this case, because we're using a glucose and a fructose, we're making a sucrose, which is one of the disaccharides. Now, can I go backwards? Yeah, I can take this sucrose and break it back into glucose and fructose through hydrolysis. And in this case, I'm going to go through a process where I'm going to end up breaking this bond. But when I break this bond, I have to restore the hydrogen and the oxygen, because remember, they're missing here when I have a disaccharide. So I'm going to have to add those back in. So both the fructose can have its full structure and the glucose can also have its full structure. Now let's talk about polysaccharides. Poly means many. These are a whole bunch of monosaccharides joined together. And remember, when we're joining them together, it's through the process of dehydration synthesis. And here's some examples. Glycogen. Glycogen is the one we really see in humans as the storage. And this is a whole bunch of glucose molecules stuck together. Now, where do we find these? Well, two of the most important places are the skeletal muscle and also liver cells. We have polysaccharides. They're covalently bonded to proteins in some cases, or lipids. So sometimes we take these polysaccharides and we add them to things. And so if in case of one of these polysaccharides bound onto a protein, we refer to it as a glycoprotein. Whereas if it's onto a lipid, it's a glycolipid. And these have a number of functions throughout the body. Here we can just see some examples of these polysaccharides. You can see each one of these is an individual monosaccharide, and you can see that it's undergone dehydration synthesis in order to allow the bonds to form. And this is really just a very convenient way of storing material. And the one we're looking at here, the polysaccharide, is glycogen. This is the one that, as I said, we find in muscles and also find in liver cells. And you can see how it forms these long branches. Now, I can come along and I can start to pull these off through hydrolysis to use them for other purposes. But it's a really good, convenient way to store a whole lot of monosaccharides. All right, we're going to end this video here. The next one will come back and start to talk about lipids.